Good afternoon, everyone. My name is Yogita, and I'm part of the content team at Renix Life Sciences. I'm the moderator for today's session. I'm excited to welcome you all to today's session. Dr. Vijay Padu will be addressing our 16th CellTalk webinar, and he is going to speak on integration of Alzheimer's disease genetics and myeloid genomics, identifies disease risk regulatory elements and genes. Dr. Vijay is working as a senior scientist at Renix, and he leads bioinformatics, genomics, and next generation sequencing research related activities in the organization. He is currently working on the neoantigen cancer vaccine projects. He holds a PhD in life sciences from TMC Attract Homi Bhabha National Institute, Navi Mumbai. His PhD study focused on cancer genomics. Previously, Vijay has worked as assistant professor at graduate and postgraduate degree colleges as well. At Trinix, he has gained experience in scientific, operational, and administrative matters. So without further delay, over to you, Dr. Vijay.
Thank you, Dr. Yogita. So as our journal club activity for CellTalk, I have chosen this research article, Integration of Alzheimer's Disease Genetics and Myeloid Genomics Identifies Disease Risks, Regulatory Elements, and Genes. The original author is Gloria Novikova. So this topic selected for Alzheimer's disease. The journal is Nature Communications, and the impact factor is 17.69. This paper was published in 2021, and this research has impact on Alzheimer's disease. So introduction, this research article, it focuses on the research done on Alzheimer's disease. Alzheimer's disease is a progressive neurological disorder, and which is commonly affecting the older people, people older than 65 years of age. There is a certain subtype which affects younger individuals also, but that is rare in numbers. So Alzheimer's disease is a neurodegenerative progression of the disorder, and it causes death of the brain cells, which results in the atrophy of the brain. And due to the atrophy of the brain, that is shrinkage of the brain, this results into the dementia onset, which causes memory decline and reduced thinking capabilities. As a result of these effects of the Alzheimer's disease, the patient's ability to behave normally and function independently in society is severely affected because the person can't remember events, his location. Even in a higher progressive state, the patient cannot recognize his own family members also. So there are certain risk factors that are associated with Alzheimer's disease. Those are old age, injury to the head, certain infections, genetic risk factors, and environmental risk factors. Now, understanding of Alzheimer's disease pathogenesis is required, but in the current state, our understanding is limited. And as right now, there are 50 million disease burden of Alzheimer's disease. That means 50 million people are affected. And in future, it is predicted that more people will be affected. So as a therapeutic point of view and also public health point of view, this is there is an urgent need to understand the pathological basis of this Alzheimer's disease. So our current understanding, it suggests the buildup of abnormal amyloid protein plaques around the brain cells as a possible cause of the disease. As the accumulation of these amyloid protein plaques, it results into the abnormal function of the neurons, and that is resulting into Alzheimer's disease phenotype. There is another theory that suggests the role of cholinergic deficits. The neurotransporter metabolite, that is acetylcholine esterase, if there is some dysfunction in its metabolism, then it can result into the Alzheimer's disease phenotype. And some studies have also involved a role of abnormal neurofibrillary tangles of the tau protein that are accumulated into the synaptic area of the neurons as a causative factor for the Alzheimer's disease pathogenesis. And there are also suggestions of synaptic damage in the neocortex and the limbic system of the brain. In a general background, there are genetic risk factors and molecular mechanisms that have been identified up till now by various research articles and groups. So the risk factors that are genetic risk factors that may play a role in the development of Alzheimer's disease, and they have been discovered over the years. So these include inheritance of variants in the genes such as APP, PSEN1, PSEN2, and ApoE. And several lines of genetic evidence have implicated myeloid cells in the etiology of the Alzheimer's disease. So these exome sequencing studies, they have identified the genes APP, PSEN1, PSEN2, and ApoE to be inherited variants and having association with the Alzheimer's disease phenotype. And recently, there have been implications of the myeloid cells also. Myeloid cells are the immune cells that are on the myeloid side of the development. That includes basophils, neutrophils, microglia, macrophages, etc. So there is evidence that they in the brain, these cells may have some role to play in the etiology of the Alzheimer's disease. Now, how these myeloid cells are implicated in the etiology of the Alzheimer's disease? So whole exome sequencing has identified many coding variants associated with the Alzheimer's disease, that is TREM23, SORL14, ABI35, and other genes, and they play an important roles in the myeloid cells of the brain, that is microglia. Then genome-wide association studies have identified common non-coding variants associated with Alzheimer's disease in more than 40 loci. So genome-wide association studies are kind of studies in which germline variants in the case and controls. Cases being the individuals which have Alzheimer's disease, and control being the normal individuals. In this study, they identify which variants, germline variants, are highly correlated with the Alzheimer's disease phenotype, and they identify those variants as risk enhancing or associated with the development of the Alzheimer's disease. So many GWAS studies have been performed, and they have identified many non-coding variants. Now, the earlier studies, they are focused on the mapping of the candidate causal genes to Alzheimer's disease risk loci using whole blood and brain expression quantitative trait loci datasets. So in the previous study, the GWAS study and quantitative trait loci of the expression of the genes in those particular risk loci that have been done. Those studies, they have used bulk cell data, that means many cells aggregate, and in that myeloid cells form approximately 10% of the total cell population. So that data is not sufficient to pinpoint which particular genes or causative variants are actually responsible for the development of Alzheimer's disease. The present challenges with respect to the knowledge about Alzheimer's disease, they include the exact mechanism of the pathological changes in the Alzheimer's disease have not been deciphered, in the sense that which genes are implicated, what is actually happening, what changes are associated, are there any certain germline variants that are associated that are leading to these mechanistic changes, that is not clear. And also there is no cure available for Alzheimer's disease. Naturally, as we don't know exact mechanism, how it is happening, so cure is not possible at this stage. The treatment available currently can only improve the symptoms. So only palliative care is available right now, but actual remedy for the Alzheimer's disease phenotype is lacking. An understanding of the pathogenesis and predisposing genetic factors are needed to devise the effective therapeutic measures. Now, this research article has had an objective that was to investigate the impact of the strong enrichment of AD risk alleles in myeloid-specific epigenomic annotations and express genes on myeloid epigenomes and transcriptomes in the modulation of Alzheimer's disease susceptibility. So, as we have seen the earlier studies, they have used GWAS data and EQTL data, but here they are integrating multiple sources of data, including GWAS, QTL sources of the data, and specifically from the myeloid cells. So they integrate it, and they see the enrichment of the Alzheimer's disease risk alleles in the myeloid specific epigenomic annotations. That means promoters and enhancers that are active in the myeloid cells. as there has been the implication of myeloid cells in the development of Alzheimer's disease. So methodology, they have done multiple experiments. So in the first, they do ChIP-seq and ATAC-seq data analysis, and they process the data for the peak calling. ChIP-seq and ATAC-seq, these are routine methods that are used to identify the chromosomes involvement or status of the chromosome in those particular cells, which part of the chromosomes are active and which parts are inactive, and which genes are actually expressing and which genes are not expressing because of the accessible status of the chromatin. So here they have identified the ChIP-seq data that are using H3K4ME1, that is the histone H3 in different methylation states, methylation 1, methylated 2, methylated 3, and H3K27AC and ATAC-seq. And this data was available for monocytes and macrophages for methylated 1 state, or monocytes and microglia for methylated 2 states, only macrophages for methylated 3 states, and all cell type for H3K27AC and ATAC-seq for all cell types. And they identified enhancers, promoters as depicted in this by using the peak calling method. The next, at the next part, they do stratification into the promoter and enhancer regions and overlap with GWAS and Hi-C data. So this data that they have with the GWAS and Hi-C. Hi-C is a method in which conformation of the chromatin is studied. Which part of the chromatin, a distant part, are coming together, actually interacting with each other. That information can be gained by using the Hi-C data. And as we have seen, GWAS data, it shows the association of the germline variants with the disease phenotype. So which of those associated germline variants are located into that particular chromatin regions, that was identified by using this study, and they stratified them into promoter and enhancer regions, those different alleles that they have identified. The next, they do de novo motif discovery in ATAC-seq regions that reside in the active enhancers in the monocyte, macrophages, and microglia. So these myeloid cells, they have their active enhancer regions, and there where exactly the transcription factors or different controlling proteins they are binding. So those particular motifs were identified in the ATAC-seq regions. ATAC-seq regions, that methodology gives you which part of the chromosome is accessible. And in that accessible regions, we know that certain risk enhancing alleles are also present. So are those alleles are affecting any particular binding motif or not, or they are disrupting actually present motif, or they are creating a new motif. That analysis was done in this particular step. Then next, they do the co-localization analysis between IGAP, GWAS, and HQTLs. These are two data sets available. This is a genome-wide association study by IGAP, and HQTL, that is histone QTLs. This gives the expression levels of those particular regions with respect to the histones that have been studied in this particular study. So to select the chromatin regions with evidence of independent or co-localized AD, GWAS, and HQTL signals. So those particular variants that are associated with the AD, Alzheimer's disease associated with the risk association, and they have co-located it with the HQTL signals to identify which particular alleles are highly expressed. The next, causal association test between the IGAP, GWAS, and the QTL datasets using SMR. Now, SMR is a method that is summary based Mendelian randomization analysis. And here they identify the correlation between the high risk of the variant with the Alzheimer's disease phenotype, and that is done by using summary of the GWAS data. And then it is correlated with the QTL data. So by using that, we get those particular alleles which are enriched with respect to the expression status. So that has been done in this particular step. And then conditional haplotype analysis to identify the association. So conditional analysis using IGAP, GWAS summary, statistic data, and ADGC, GWAS cohort, individual level genotype data as a reference panel. So they included these two data sets, IGAP, GWAS, and ADGC, GWAS cohort as a reference panel, and they did this conditional and haplotype analysis to identify association of those particular variants with respect to the disease state. The next step in the methodology, they perform prioritization of the candidate causal variants. So they have found thousands of different variants to be associated with the Alzheimer's disease phenotype, as these are genome-wide. The genome is a very large entity, so there are millions of different variants that are there. So some thousand come up as a statistically significant that are associated with the Alzheimer's disease. Now some prioritization needs to be done, and they have used these steps to prioritize that. So they selected the variants that reside in the active enhancers of the monocyte macrophages and microglia. These are the myeloid cells. So as the study is focusing on the myeloid cells, so they identified those variants that are residing in the enhancers and promoter regions of these microglial genes. And then motif disruption creation analysis on the selected variants. Those variants that have been highly associated with the Alzheimer's disease phenotype, are they disrupting existing motif of the nucleotide sequences that are involved in the protein binding, some transcription factor binding, or they are creating the some transcription factor binding motif into the genome? That was the second criteria. Then the remaining variants were screened for the eQTLs in the monocytes and macrophage. That means whatever variants that were selected up to the second step, then the next step was the eQTLs. eQTLs provides the expression quantitative trait loci. So at that particular stage, they screened for this eQTL expression status in monocytes and macrophages. And in this way, they prioritize the candidate causal variants. The next, transcription binding motif disruption and creation analysis. Again, as in the step 2, they identified those particular location of the variants, and they studied if they are disrupting pre-existing transcription binding motif or they are creating anything new, so that it will affect the gene expression. Then at the next step, they generated the patient-derived human-induced pluripotent stem cell microglia for ATSC-seq and RNA-seq analysis. And they use this HIPSC microglia for the validation of one of the variants that they have discovered by using ATSC-seq and an RNA-seq analysis. And at the next step, they do allele-specific expression and open chromatin analysis using the RNA-seq data. That means the RNA-seq data will provide the expression level of each and every gene that is expressing that particular cell type, and they correlated that data with the position of that particular allele. So if in that particular position of the allele, if it is expressing, then RNA read will align to that particular location. If that is not expressed, then RNA read will not align to that particular location. So they check whether that particular allele is getting expressed or not. So coming to the results, so here they show that the AD risk alleles, Alzheimer's disease risk alleles, are specifically enriched in myeloid active enhancers and in putative transcription factor binding sites located in these enhancers. So we have 3 panels. First is the monocyte, then macrophages and microglia. And there are different stratified genes, as we have seen earlier, that they are stratified in different regions. So this is the active enhancer, active promoter. Then this is an enhancer that is primed enhancer, and this is the primed promoter. So active, that means it is actively getting expressed, and primed means certain assembly of transcription factor is ready in that particular position, but it is not actively transcribed. Now, what does this result show is that this dashed line is the Bonferroni corrected multiple correlation analysis. So p-value, this is the corrected p-value. So as we can see that in monocytes, the active enhancers that are related to the Alzheimer's disease, that is shown in this purple, and they have compared it with the schizophrenia also, that is shown in gray. So these active enhancers in monocytes, active enhancers in macrophages, and active enhancers in microglia, they are statistically significantly enriched in this study analysis. In the next panel, they find the transcription factor binding motif and the log10 p-value of LDSC, that is LD score analysis, enrichment analysis. And here, as we can see, there are certain genes that are moving ahead of the normal p-value threshold. So here there are different genes that are statistically significantly associated. So TGF, PU1, Here also these two genes and these 4 genes and 3 genes here. So these genes they find to be statistically significantly enriched in the myeloid active enhancers. In this way, they find different genes as a result of integration of Alzheimer's disease GWAS signals with myeloid epigenomic annotations and chromatic interactions by using promoter capture Hi-C and eQTIL datasets, and they find 16 AD risk loci. So these are the different risk loci, and those highlighted in bold, these are associated with the disease risk. So as you can see, monocyte and monocyte derived macrophages, some common genes are also present in both the sets. So these are the different gene locus. Locus means that particular gene and the surrounding genes. And here we can see that in BIN1 that have been strongly associated with the disease phenotype. And in this SPI1 locus, these genes have been found to be associated, but those in the bold, they are found to be highly associated with gene expression and the disease risk. So in this way, they have found different alleles, and this is a result of integration of GWAS analysis and also the myeloid cell epigenetic analysis. Next result shows that AD risk enhancers spatially interact with the promoters of BIN1 and RAB-EP1 and regulate their expression in the myeloid cells. So these are some candidate genes that have been identified here. This is the BIN1 and RAB-EP1. So here they have studied this in detail. So these are the two panels showing studies for RABP1 gene locus, and first panel showing the results for the BIN1 locus. Now this is the GWAS data, Alzheimer's disease GWAS data, and here the y-axis shows the log10 p-value, and the x-axis shows the genes, that is the location of the genes in this particular locus, BIN1 locus, and here RABP1 locus. So those points that are shown in red, these are variants which are statistically significant. That means they have very low p-value. Therefore, their log10 p-value is higher. Here also, those associated with higher disease risk, those variants are shown in red. And as you can see, they are enriched in this particular region. And this is where the BIN1 gene is located. That is depicted in red. So this BIN1 gene, it shows the direction of the transcription. Now here, these red bars, they show the actual binding sites of located in these regions, and the height shows their statistical significance. Here, it shows the interaction. This is from the Hi-C. Hi-C capture, it gives you the association between different chromatin region, and this line, these curves, they depict that these regions are actually physically interacting with each other, and depth of this curve, which shows how significant that particular interaction is. So here they also find in this method also that there is a physical association between these regions. Here they have carried out another Hi-C study. This is the study in the monocytes, and this is in the macrophages, and by using this both two cell types, they get the same result. So that means this association is verified by using two different methods. And same is done with RABP1. So there is also a region, binding region, that is statistically significant and is found to be interacting with the enhancer of that gene. So in the same way, we can see in the macrophages also there is a statistically significant interaction between the enhancer and the binding region, the promoter binding region of that particular RABP1 locus. Now in the next result, they identify putative causal association between chromatic activity, target gene expression regulation, and Alzheimer's disease risk modification point to candidate causal genes in the myeloid cells. So here, the genes that are depicted in the blue, purple, and red, these are statistically significant genes that have been identified. The y-axis here shows the log10 of p-value. So higher bars represent higher statistical significance. And they find that different loci, as you can see, there are different disease loci, gene loci, and the neighboring genes. There are certain genes that are found to be significantly associated with the chromatin activity, target gene expression regulation, and Alzheimer's disease risk modulation. Now here, they give us the candidate causal genes identified through the integration of ADGVAS signals with myeloid active enhancers and annotations, HQTL and EQTL datasets. So histone QTL, expression QTL, that gives the expression level and the epigenetic status of that particular locus, and those genes that are associated with the those particular enhancer regions active in the myeloid cells, and integrating that with the Alzheimer's disease genome-wide association studies. So those risk alleles that are located in these gene locus have been found to be statistically significantly associated with the epigenetic activity in that particular region. So they have shortlisted this. They have started with some 20,000, 30,000 genes initially, and as a result of this analysis, they have shortlisted these genes. Now here, they study one particular gene, that is MS4A6A in MS4A locus, and they validate this gene in a human pluripotent stem cell microglial cell. And in this study, they show that this particular variant that is enriched in this particular gene, it disrupts and anchors CTCF binding site. So CTCFs, these are important in the maintenance and creation and disrupting of the chromatin structures that leads to changes into gene expression. So this CTCF binding is required for proper functioning, and that is done by bringing different locations of the chromosome together. So here they show that this particular variant that is found in this gene, this locus and this particular gene, it is disrupting that CTCF locus. It is resulting into the increased expression of that particular target gene. So to understand this panel, there is this locus where highly enriched alleles are there. And then there is the data from ChIP-seq in which those particular binding transcription factors, these are binding in this particular location in this region. Again, data from another cells, microglia, another technique, ATAC-seq and microglia. These genes are accessible in the chromatin. The genes that are located, multiple genes that are located in this particular locus, and the candidate gene shown in the red. And further, they see the interaction between this CTCF loop and the promoter of this gene, MS4A6A, and they show that there is an interaction. The interaction has been proved by using monocytes and macrophages in two ways. And then they do ChIP-seq analysis, and in that also they find that these two locations, that is anchor CTCF binding loops, they have an interaction. And further, they also show that in a B lymphoid cell line by using another technique, that they show that these two sites are also interacting. Next, in the second panel, these are the gene loci in the MS4A locus, and this red is the variant, location of the variant that is highly associated with this Alzheimer's disease risk locus. Now this variant is normally present here. So C is the nucleotide present commonly, but this variant, it has T in that particular location. So they studied what happens in these two situations when the C is present and when T is present. So in that case, they did that study in microglial cell, the human HIPSC derived microglial cell, and they studied these different markers in those cells to prove that these are indeed developed in microglial cell type. And in that, they analyzed this ATAC-seq read counts. So as you can see, the C is depicted in blue, the normal allele, and the T allele, that is variant associated with the Alzheimer's disease loci. So here, ATAC-seq read counts, these are more in case of C alleles and less in case of T alleles. Here also we can see that ATSC rates are more in case of C alleles as compared to T alleles. So that means chromosome is more accessible when C allele is there and less accessible when T allele is there. So what it is doing is that when instead of CT is there, this CTCF binding site, it is getting disrupted. And due to that, the expression level that would normally be less, shown here in case of blue, it is increasing. So this particular gene expression is getting increased in this case. So in this way, they have validated this variant located in this MS4A6A gene, that this particular variant, T variant, it is disrupting the CTCF binding site, and this is resulting into increased expression of that gene. Now, this last result, it shows a Manhattan plot. Manhattan plot shows the what are the risk associated alleles that are present in the GWAS data. So here, this red genes that are depicted, they indicate that increased expression of the gene is predicted to increase the risk for Alzheimer's disease. In blue, the decreased expression of this particular alleles is predicted to increase the risk of Alzheimer's disease. And those in gold, there is no definite direction identified where this particular expression will lead to the either will increase the Alzheimer's disease or not. So they have identified this by using high C and SMR approaches. So going to the discussion, this study reports an integration of Alzheimer's disease GWAS with epigenomic and transcriptomic data sets from myeloid cells to nominate candidate causal variants and regulatory elements and genes and pathways. So thousands of variants that were presented by GWAS to be significantly associated, they have been shortlisted to a small list and that are associated with the myeloid cell epigenetic activity. Then many of the candidate causal genes that were identified in myeloid cells, they are functionally related to the endolysosomal system. This endolysosomal system is important in the integration of the unused cellular material and recycling it and providing it for the recycling of that particular material. So this results indicate that that activity is getting affected in case of this myeloid cells and Alzheimer's disease risk alleles interaction. Now, through this integration, the study informs a mechanistic understanding of Alzheimer's disease genetics and pathobiology for the formulation of novel therapeutic hypotheses. So by integrating these different data sets, they are shortlisting those particular genes and also coming up with a variant, and they have also validated one variant, and there are other variants of which the mechanistic pathways can be deduced, but they have given a list of the genes that may play some significant role in the pathobiology of this particular Alzheimer's disease. Then integrative genomic approaches present in the study offer a framework to identify regulatory elements, genes, and variants that are likely causal for Alzheimer's disease. Now they have used different dataset GWAS and epigenetic datasets. Other datasets can also be created and they can also be integrated, but this study offers a framework how to integrate these different datasets in one study. Conclusion, the study reveals a link between chromatin activity, gene expression, and Alzheimer's disease risk in myeloid cells and proposes the molecular mechanism of action of candidate functional variants in several Alzheimer's disease risk loci. The study identifies specific Alzheimer's disease risk enhancers that are burdened by these variants and regulate target gene expression. The identified specific Alzheimer's disease risk enhancers most likely modulate disease susceptibility by altering the biology of the myeloid cells. The study highlights the coalescence of candidate causal genes in endolysosomal system of myeloid cells and underscore its importance in etiology of Alzheimer's disease. The impact of the research, the study uncovers a mechanistic understanding of Alzheimer's disease genetics and pathobiology. This mechanistic understanding will be useful for the formulation of novel therapeutic hypotheses. Thank you for patiently listening.
Thank you so much, Dr. Vijay, for an enlightening and insightful talk. Now the audience is welcome to have questions. Dr. Shaheen Shah has a question. What is candidate causal genes?
Okay, so candidate causal genes, those are the genes in which GWAS studies, genome-wide association studies. Now these studies, they find association of different germline variants with the Alzheimer's disease state. So in those studies, they find which allele is associated with the Alzheimer's disease phenotype as compared to the normal individuals which do not have Alzheimer's disease. So in that, they identify those particular alleles, and candidate causal genes are the genes in which those alleles are located.
Hope it answers your question, Dr. Shaheen. We have a question from Dr. Nupur. The researchers identified several new candidate genes. Out of them, they studied MS4A locus disrupted by CTCF. What about other candidate genes? They also may have other implications.
Yes, definitely. There are multiple genes. They have identified 26 genes in the first step, then they shortlisted 16 genes. But for one research article presenting all the things will not be feasible. So maybe in the future they will try linking other genes. But they have conclusively proved that myeloid epigenetics and the association of the variants identified by GWAS studies, that has been established in this study.
Yeah, it's definitely a nice paper and nice presentation also. Yeah, they studied only one gene. Actually, yeah, that's why I was thinking whether this particular gene is already a risk gene.
They have identified this risk gene through their integrative analysis. They have come up with this particular gene. And as this is a myeloid gene, so they have studied in the microglia, which is also a myeloid gene. And therefore, they have found out that it is disrupting a CTCF binding loop, which is resulting into the expression changes. So at that particular point, so what they I think proving one gene gives them a foolproof thing that whatever they are claiming is actually happening, at least with respect to one gene. Okay, so other genes they can definitely study.
Okay, thank you.
Does anyone have any questions or opinions regarding the presentation? I think there are no questions. So thank you again, Dr. Vijay, for the great presentation. It was pleasant to hear such an informative talk. Dr. Vijay, could you please go to your final slide? Thank you. I would like to inform you about our next presentation. The next presentation will be held on 11th October 2022. The next presented will be Dr. Shaheen Shah, and she will deliver a talk on triggers and elevating factors for fatigue in Parkinson's disease. So thank you all for being here. I appreciate your presence and kind attention. Hope to see you all at the next presentation. Many thanks.